Hello everyone, my name is Eduardo Lamas, and in this video we're going to talk about the process of phagocytosis. The first step in phagocytosis is the chemotaxis and adherence of the microbe to the cell. Then the cell is going to ingest a microbe inside a phagosome. This phagosome is going to fuse with the lysosome to form a phagolysosome, the most important compartment in this process. The next step is the formation of the residual body, which contains indigestive material. This material is going to be discharged by the cell. Polymorphonuclear cells, or PMNs, and macrophages contain multiple lysosomes and granules for PMNs that are used in preparation for microbial killing once there is fusion with the phagosome. Primary and secondary granules fuse with lysosomes and phagosomes to form the phagolysosome, in which microbial killing takes place. An NADPH oxidase enzyme complex is formed in the membrane of the phagolysosome, guided by the protein RAG2. This NADPH oxidase is required for the generation of superoxide anion, an oxygen radical that participates in destroying the microbe. Other enzymes within the phagolysosome utilize superoxide anion as a substrate to generate other free radicals. For example, superoxide dismutase in an acidic pH, such as that found in the phagolysosome, can convert superoxide anion to hydrogen peroxide. These reactive oxygen species, also known as ROS, and hydrolytic enzymes will degrade the bacteria and then release the debris via exocytosis. Therefore, NADPH oxidase is a major source of toxic reactive oxygen species. Macrophages have different mechanisms by which they can kill microbes. Some of them include antimicrobial peptides, such as cathelicidin, macrophage elastase-derived peptide, toxic oxygen-derived products, including superoxide anion, hydrogen peroxidase, singlet oxygen, hydroxy radical, and hypohalite. Another mechanism is formation of nitrogen oxide, which is a vasodilator. In addition, acidification to a pH of about 3 or 4 has bacteriostatic or bactericidal properties. Finally, enzymes including lysozymes, which digest the cell walls of gram-positive organisms, acid hydrolases such as elastases, which break down ingested microbes, are also used by macrophages to complete their phagocytic function. Oxygen-independent killing is also present in the phagolysosome. For example, lysozymes can hydrolyze mucopeptides found in the cell wall. Lactoferrin will deprive pathogens of iron. Proteases will digest killed organisms by breaking down proteins. Finally, antimicrobial peptides, such as defensin, will lead to lysis of the microorganism. Here we're going to go into a little bit more detail about reactive oxygen species, also known as respiratory bursts, because this is a very essential mechanism used by cells of the immune system in the killing of microbes. For example, oxygen-dependent mild or peroxidase-independent reactions. In this case, we can see how glucose is interacting with NADP, a reaction that is catalyzed by glucose 6-phosphate dehydrogenase to generate pentose phosphate and NADPH. This NADPH is going to react with oxygen, a reaction mediated by NADPH oxidase and cytochrome B558, to generate NADP and superoxide anion. This superoxide anion molecule is going to react with hydrogen, catalyzed by superoxide dismutase, to generate hydrogen peroxide and singlet oxygens. In addition, superoxide anion can react with hydrogen peroxide to produce the very potent hydroxy radicals and singlet oxygen. Respiratory burst also includes oxygen-dependent myeloperoxidase-dependent reactions. For example, hydrogen peroxide can react with chlorine mediated by myeloperoxidase to produce hypochlorous acid and water. This hypochlorous acid can further react with water to generate singlet oxygens, chlorine, and water. Another reaction included in respiratory burst is the detoxification reaction. In this case, we can see how hydrogen is interacting with superoxide anion catalyzed by superoxide dismutase to generate hydrogen peroxide and oxygen. This hydrogen peroxide can be catalyzed by catalase into water and oxygen. It is important to note that catalases are used by certain organisms to avoid killing. By doing this, they convert the potentially damaging hydrogen peroxide into water and oxygen and thus avoid killing. When finished watching this video, please help us determine if it was helpful to you by using the thumb up or down option below. Thank you.